So let's get to the meat of it, to the neuromuscular junction, okay? So here what we can see in this picture, right, the purple ones are the muscle fibers, okay, coming down. In the dark stain, we have this axon coming down, right, from that, from the spinal cord. The cell body is in the spinal cord, remember, and this axon is leaving the spinal cord and going and traveling towards the muscles. Can be that extensor, flexor, whichever muscle we want to focus in the skeletal system, right? And these motor axons will then end up, you know, on these fibers. You see the fiber as motor end plates. So that's kind of a $10 name for the neuromuscular junction. Where it is happening at the motor end plate, okay? And that's, again, a chemical synapse, right? There is a neurotransmitter that is a chemical between this efferent neuron and the muscle fiber that is going to be receiving those neurotransmitters, okay? So just this term that we introduced, the motor end plate, right, is kind of a $10 word for neuromuscular junction, okay? So we don't get confused if the question is talking about motor end plates. We are actually focusing on the axon, presynaptic terminal, and the muscle as a postsynaptic membrane of a neuromuscular junction. And here is what we are going to now do. We are going to introduce the first neurotransmitter of our journey in the neuroscience, okay? And this one is called acetylcholine. Remember all those chemicals in synaptic vesicles, you know, one of them is called acetylcholine, okay? And it was first discovered in this neuromuscular junction, and it is very well studied because of this, of the kind of approachability, so to speak, at the neuromuscular junction at the periphery, okay? So basically, the neurotransmitter is a chemical message, right, in specific, with specific characteristics, you know, that is released from this presynaptic motor axon, you know, and is looking for its receptors on the muscle fibers, okay? Once it was discovered in the neuromuscular junction, further studies also revealed that acetylcholine is also present in the central nervous system, okay? And we're going to talk about them when the time comes. Today, we are focusing only on the neuromuscular junction, okay? Again, in this upper right corner, you can see muscle fibers, you know, these axon terminals with these motor end plates, right? And now if we zoom in, we are already familiar with this. We know it. Okay, so we're just going to give some names to it. Okay, again, we have this presynaptic terminal. You see this axon coming down, right? It has these calcium channels, right, in this presynaptic terminal. It has these synaptic vesicles. And in this case, these red dots, they are neurotransmitters, but now we know the name, right? It's acetylcholine because we are talking about neuromuscular junction. In this case, you can see we can even see this myelin sheath, the Schwann cells, because remember the cell body of this motor neuron is inside the central nervous system, but the axon is at periphery. So who will provide the myelin for the lower motor neuron or efferent neuron will be Schwann cell, okay? So let's not confuse them. Even though cell body is central nervous system, the myelin is provided at periphery. So it is a Schwann cell that is giving that myelin insulation for this presynaptic terminal that is trying to reach the muscle, okay? So now let's walk through it. And you already know it, okay? You will see how much you know. So basically the action potential is coming down, remember, through this axon, is changing the voltage, right, of the membrane. The calcium channels will open, which means the calcium that is outside and based on its electrochemical gradient, it will rush in. This calcium will go in and move the synaptic vesicles with acetylcholine, right, towards the membrane, right? And this acetylcholine will be released into the synaptic cleft, and acetylcholine will now search for its receptors. You see on this muscle as a postsynaptic membrane, right? And this kind of binding of acetylcholine to the postsynaptic receptor, the acetylcholine receptor site, will allow the sodium to come in and depolarize the membrane of a muscle, okay? So not to overwork the muscle, we have to get rid of acetylcholine, and here is that enzyme. You remember that one that is cutting the acetylcholine in two pieces, acetyl and choline. That's why it's called acetylcholine. And this enzyme is called acetylcholine esterase. Okay, so and this is now finely tuned system. So basically, action potential comes in, acetylcholine is released, and then acetylcholine is chopped off. Okay, and certain number of these acetylcholines have to bind certain number of receptors that the muscle has its strength. Okay, if you have 100 acetylcholines, the strength of the muscle will be certain level. If you have 1,000 acetylcholines, the muscle strength will be higher, okay, because more of this acetylcholine will bind more of the receptors. And as you can see, even in this case, you have 1 channel open, 2 channel open, 3 channels open, right, that allowing sodium in, but 1, 2, 3 are still closed. So if more acetylcholine comes in, more of them will be open, more sodium will come in, the higher shift of the membrane potential will be achieved towards the threshold so that the muscle can actually contract, okay? All this together is again called motor end plate or neuromuscular junction, okay? So where is that neuromuscular junction formed? If you look at the top, right, we have this axon, we have this myelin with the Schwann cells, the axon terminals are coming in, and here is a new term or new anatomical kind of part or component that we have to learn is the sarcolemma, okay? So that's a new term that we have to learn about neuromuscular junction, and that sarcolemma is actually a cell membrane, okay, which surrounds or these muscles, these striated, fibers coming in, okay? And you can see here, when we zoom in on this, in this mid kind of panel, the sarcolemma is here on the surface of this muscle, right? And the receptors are actually inside this sarcolemma. Sarcolemma is actually a membrane, okay, with these channels in awaiting the acetylcholine to bind to allow the sodium to come in when it is bound. You can see the example, one acetylcholine receptor that has bound these 2 circles in this lowest panel, right, will allow sodium to come in, while the one receptor will be blocked or closed, and the second receptor will be blocked or closed because no acetylcholine is bound, okay? And just a reminder, that's a chemical, a neurotransmitter, that is released by a nerve to bind to the postsynaptic terminals, okay? First disease affecting the neuromuscular junction, okay, that we will learn about is myasthenia gravis, okay? And here is what is the most important or giveaway symptom when a patient walks into your office, okay, and says, doctor, this and this, when I do something, my ability to perform that action, gets worse as I do it or repeat it over and over again. And then if I take a rest, right, it improves. It gets better. And then again, if I reuse it, it gets worse and worse and worse. One example could be, let's say, blinking, right? First blink, it's very easy, and then if I want to make second and third blink, it's much harder and harder to blink, okay? And then if I take a rest for an hour, I can again blink normally first time and then worse and worse and worse, okay? So that's very characteristic of myasthenia gravis, very specific for this disease. And now we're going to kind of try to understand why is that the case, okay? So basically, myasthenia gravis is considered to be autoimmune disorder. Okay, what does that mean? That our own protectors, right, that are protecting us against viruses, against bacteria, against any intruders, right? Our own antibodies, right, that are floating around will start attacking something in the neuromuscular junction. Okay, so what is it? Okay, in this case, for some reason, the body in this patient will produce an antibody against acetylcholine receptor. Okay, in the left side, we can see the normal situation, right? We have this presynaptic terminal, acetylcholine in synaptic vesicles, the calcium comes in, it is released, and now all this acetylcholine can freely bind, to the receptors that it can reach, right? And this will change the membrane potential, and contraction of the muscle cell will happen. But in the case of myasthenia gravis here on the right, what happens is antibodies will go and bind the receptors. And now if antibodies are blocking the receptors, where will acetylcholine bind? It cannot bind. It cannot allow sodium to go in, right? All the receptors, or many of them, are blocked, okay? So, and blocking of these receptors is actually preventing, acetylcholine to induce, you know, the conformational change of the postsynaptic receptor to allow sodium to come in. And if sodium is not coming in, there is no change in the voltage, right? There is no depolarization. And for that reason, the muscle cell cannot contract, and that's known as paralysis. Okay, here is one example how paralysis happens, and you now know the concept or mechanism how it is happening, because there are antibodies blocking the receptors on the sarcolemma, on that membrane, right, of the muscle. All right. Okay. So what are the symptoms? The first symptom is the eye muscle weakness. You can see here, you know, how this patient basically on the left eye cannot even keep eye open because it cannot stimulate, right, this muscle to open the eye, right? There is no depolarization of the muscle to contract to keep this eye open. But this is not the only case, right? Any muscle that we use for a movement, right, that can contribute, let's say, to eye movement, left, right, up and down, to smiling, to chewing, to talking, to swallowing. So wherever we have a muscle, you know, that is kind of being actively used, right, these receptors will be attacked. Wherever there is a neuromuscular junction, so to speak, you know, and then kind of the ultimate problem is if the muscles for breathing are affected, you know, these receptors for acetylcholine on the breathing muscles are being attacked, you know, it's harder and harder to breathe, and that can be then life-threatening or life-dangerous, okay? We are able to actually, patient is able to move first normally and then actually kind of wanes off, is when this acetylcholine actually comes out, right, and seeps into the synapse of the patient. We still have some receptors available. You see these ones on the left, right, available, and some in between, okay? Which means the first set of acetylcholine that will come in, right, will be able to bind these receptors that are available and induce the initial set of contractions, okay? However, you know, to remove them from the synapse, right, you remember we need the enzyme to work, we need glia cells to work, some reuptake to happen, and so on. So it takes time. So that means that all the other acetylcholines that are now coming maybe down with the second action potential, because we want to move for the second time, right? They don't have any more receptors where to bind, okay? And the second one will be worse and worse and will be harder and harder to initiate that movement, okay? Because the available receptors are taken by first set of acetylcholine and by the autoantibodies, okay? So there is no more free receptors or sodium channels to open to allow influx in, to allow second kind of contraction of the muscle, okay? So hope that's clear. So that's why in this case, the movement actually worsens after the periods of activity. And then if the patient is taking the rest, it is actually allowing the time to remove the acetylcholine from the initial kind of rush or initial blast, right, by the enzyme or by the diffusion or by the glia cells. Okay. And now after some time, we have cleared out, so to speak, the ones that are available, and next action potential can come in and induce that contraction. Okay. Okay. So